Hello and welcome students. I, Dr. Adil Patel, welcomes you for the discussion of the topic distribution, tissue permeability of drugs and binding of drugs to various tissues. What is distribution? So, the distribution is defined as the reversible transfer of a drug between one compartment and another. So for the ease of the study, the entire human body is divided into various compartments, and it has been believed that the drug is getting distributed between all these hypothetical body compartments. The drug distribution refers to the movement of drug to and from the blood and various tissues of the body, for example, fat, muscle, and brain tissue, and the relative proportions of the drug in the tissue. In this figure, you can see how the movement of drug takes place inside the body, or you can say how the drug gets distributed between various compartments of the body. So once the drug is distributed in the body, it can go for the drug metabolism. After drug metabolism, the drug can go for the excretion. Some amount of drug directly goes into the excretion without getting metabolized, and in between you can see that there is a central compartment called the plasma compartment. So the blood can enter into plasma compartment once it is administered to the body, and even after metabolism, some amount of drug which remains unmetabolized can enter into the blood plasma. From blood plasma, drug goes into the excretory organ, and from these particular organs, drug is being excreted from the body. Now there are two major rate limiting steps in the distribution of drugs, and that are rate of tissue permeation and the rate of blood perfusion. Now let us see the physicochemical properties of drug and how it affects the distribution. So a very important physicochemical properties of drug that influence its distribution are molecular size, degree of ionization, partition coefficient, and stereochemical nature. So these properties are affecting the rate and the extent of distribution of drug inside the body. Almost all drugs having molecular weight less than 500 to 600 Daltons easily cross the capillary membrane to diffuse into the extracellular interstitial fluids. So low molecular weight drugs are having great access compared to high molecular weight drugs. Now only small water soluble molecules and ions of size below 50 Daltons enter the cell through aqueous filled channels, whereas those of larger size are restricted unless a specialized transport system exists for them. So in previous lecture we had discussion about the active transport mechanism and the facilitated or the carrier mediated transport mechanism. So that is applicable here where the drug molecular size is greater than 50 Dalton. The degree of ionization of a drug is an important determinant in its tissue permeability. So whether the drug is available in the ionized form or in unionized form, that also affects the tissue permeability of the drug. A drug that remains unionized at physiological pH values can permeate the cells relatively more rapidly, because if you see the structure of cell walls are predominantly lipophilic in nature, and this type of nature, that is lipophilic nature, is more suitable for unionized drug. All drugs that ionizes at plasma pH, for example polar hydrophilic drugs, cannot penetrate the lipoidal cell membrane, and tissue permeability is the rate limiting step in the distribution of such drugs. So drugs which are highly polar and water soluble, so they are facing difficulties while crossing or moving through the lipoidal cell membrane, and in case of such drugs the permeability of drugs is the rate limiting step for the distribution. Now only unionized drugs, which are generally lipophilic, rapidly cross the cell membrane. So that is what we have learned from previous 2 points, that drug which remains unionized at physiological pH has greater access compared to the drug which goes into the ionization phase. Now this figure would help you to understand the movement of drug across the cell membrane. So as you can see, the unionized drug with low partition coefficient and large oil water partition coefficient can easily cross the capillary wall and the cell membrane, whereas the ionized drug can easily cross the capillary wall. The pores present in the capillary pore will facilitate their entry, but unfortunately they will not be able to cross the cell membrane. So the permeation of unionized and ionized drug across the capillary and the cell membrane has been shown in this figure. Now what happens in case of polar drugs, where permeability is the rate limiting step in the distribution? The driving force is the effective partition coefficient of drug. Please remember this term, effective partition coefficient of drug. How can we calculate the effective partition coefficient of drug? So the equation is fraction of unionized drug at the pH 7.4 into the oil water coefficient of unionized drug. So that will give us the effective partition coefficient of the drug. And as it has been mentioned here, that it is a very important parameter to decide the tissue permeability of ionized drug. Now let us see the physiological barriers to distribution of drugs. So a membrane or a barrier with spatial structure features can be permeability restriction to distribution of drugs to some tissues. Let us see what kind of physiological barriers we have inside the body. Number 1, simple capillary endothelial barrier, simple cell membrane barrier, blood brain barrier, blood cerebrospinal fluid, that is CSF barrier, blood placental barrier, and the last one is the blood testis barrier. Now let us try to understand about each physiological barrier in brief. So starting with the simple capillary endothelial barrier. So all drugs, whether ionized or unionized, with a molecular size less than 600 Daltons, diffuse through the capillary endothelium and into the interstitial fluid. Only drugs bound to the blood components are restricted because of the large molecular size of the complex. So later we will discuss about the drug protein binding. So in case of drug protein binding, the drug may not be able to cross the simple capillary endothelial barrier. The next is simple cell membrane barrier. So drugs entry into cells of most tissues is limited by its permeability through the membrane that lines such cells. So the outermost lining of the cell is called cell membrane. Such a simple cell membrane is similar to the lipoidal barrier in the gastrointestinal absorption of drugs. So when we talk about the absorption of drug from the stomach or from the gastrointestinal tract to systemic circulation, the most important barrier here is the simple cell membrane barrier, which is predominantly lipophilic in nature. The next barrier, physiological barrier, is blood-brain barrier, which is also known as BBB, and this is a very important protective barrier around the brain, and it will restrict the entry of foreign material as well as component directly to the brain. The capillaries in the brain are highly specialized and much less permeable to water soluble drugs. The brain capillaries consist of endothelial cells which are joined to one another by continuous tight intercellular junctions comprising what is called as the blood-brain barrier. So a very tight intercellular junction of capillaries is responsible for the making of blood-brain barrier, and it is highly specialized barrier and much less permeable to water soluble drugs. Now the presence of spatial cells called pericytes and astrocytes, which are the elements of the supporting tissue found at the base of endothelial membrane, from a solid envelope around the brain capillaries. So a solid envelope kind of structure is made up by two types of specialized cell called pericytes and astrocytes. As a result, the intercellular, which is also known as the paracellular passage, is blocked, and for a drug to gain access from the capillary circulation into the brain, it has to pass through the cells, which is transcellular route rather than between them. So intercellular space is blocked and every drug has to pass through the cell, which is intracellular route, and that is why the blood brain barrier is having very limited access to many water soluble drugs. Now a solute may thus gain access to brain via only of two pathways. So there are only two possibilities where the drug can enter into the brain via the blood brain barrier. Number one is passive diffusion through the lipoidal barrier, which is restricted to small molecules having high oil water partition coefficient. The second pathway is active transport of essential nutrients such as sugars and amino acids. Next is blood cerebrospinal fluid barrier. So the capillary endothelium that lies the choroid plexus have open junctions or gaps, and drugs can flow freely into extracellular space between the capillary wall and the choroidal cells. So again, this is a very highly specific physiological barrier, and it mainly available, or it is mainly present in the spinal cord region. However, the choroidal cells are joined to each other by tight junctions, forming the blood CSF barrier, which has permeability characteristics similar to that of the blood brain barrier. The next is blood placental barrier. So many drugs having molecular weight less than 1000 Daltons and moderate to high lipid solubility cross the barrier by simple diffusion quite rapidly. This shows that the placental barrier is not as effective as barrier as blood-brain barrier. So the blood placental barrier is very generous when it comes to the movement of drug, and it is not as restrictive and specific compared to the blood-brain barrier. The last one is the blood testis barrier. So it is the tight junction between the neighboring Sertoli cells that act as the blood testis barrier. Now this barrier restricts the passage of drugs to spermatocytes and spermatids. So the soft organ which are available or which are present in this region, so the entry of drug to these particular organs are mainly restricted by the blood testis barrier. Now coming to the concept of organ, tissue size and perfusion rate. So the distribution will be perfusion rate limited when the drug is highly lipophilic and the membrane across which the drug is supposed to diffuse is highly permeable, such as those of the capillaries and the muscles. The perfusion rate is defined as the volume of blood that flows per unit time per unit volume to the tissue. So basically it is the rate and the amount of blood passing through a particular organ or a particular tissue. It is expressed in ml per minute per ml of the tissue. Now the extent to which a drug is distributed in a particular tissue or organ depends upon the size of the tissue, for example tissue volume, and the tissue blood partition coefficient of the drug. So dear students, that was all about the drug distribution, various physiological barriers, and how the movement of drug is taking place inside the body and how various physiological barriers and organs are affecting the distribution of body. Please go through this video and let me know if you have any query or question. Thank you very much for joining. Have a good day.